Okay, so today we're going to be talking about some more advanced topics in development, and it's kind of like a potpourri of things, but all interesting. So we'll start off with some developmental neurobiology experiments to sort of try to manipulate development and get some idea of what some of the constraints on development are, particularly development of cortical areas. And then I'll talk about gyrification. So we know that we have lots of folds in our brain. How do they get there? What's sort of the general theory of why there are folds in the brain? And in particular, now we'll talk about something called allometry. You probably haven't heard of that, but that's basically the kinds of relationships. What are the rules for how the sizes of things change as you change the overall size of the body? So there's certain very characteristic things that happen as you scale up the body because you change surface area and volume differently and things like that. So we'll talk about that in relationship to gyrification. And then last, we'll talk about the subplate. So the subplate is something that happens in mammals. And it's particularly involved in the formation of cortical maps. And so we'll talk about what does that look like. It's not a cortical plate, meaning like the whole thickness of the cortex, but it turns out there's this thing called the subplate, which is even bigger than the cortical plate, the final product in development. Okay, so we'll start off with three quick and dirty experiments, developmental experiments. And so the first one that I often talk about is chicken quail grafting experiments. So chicken, quail. So chickens and quails are pretty embryonic graft. So chickens and quails are actually very similar to each other, and it turns out you can actually transplant parts of the brain between chickens and quails. And one of the ideas is just to try to get some idea of, at what stage is a quail-specific or chicken-specific specification of some kind of behavior actually crystallized. Because obviously the problem with development is you start off with a single cell, the egg, and then it divides, and then gradually the different offspring go their own ways. And now they've all got the same exact genetic material in there. It's controlled differently. But one question is, at what point does one of the offspring become specified to sort of do a specific thing different from the other offspring? And so these experiments are often they're often done in chicken quail eggs, because in comparison to a mammal, you can sort of get into so here's the fourth ventricle, which you can already see in the embryo. And so you can sort of operate on an egg, on the embryo on an egg, and then grow it up. It's easier than sort of trying to mess around with something in the actual uterus, like take it out, put it back in. That's actually been done too. But what these experiments were is you take a little, and I always say this when you're doing these experiments, compared to doing something with electronic circuits, it's a little bit more crude. So what these experiments have done is you take a little chunk of the brainstem, say the motor part of the brainstem, and then you make a little hole over here. So say this was, which way do I have it set up? Say this was a chick, and this was a quail egg. So basically you just take a little bit of this guy over here, make a hole, and throw that little piece out. So here's that little piece. We cut it out, and throw that out, and then take this piece and put it in there. So, like I said, we're not talking rocket science. We're not exactly taking out the same piece, close enough, and we're not taking out exactly the right piece. But it turns out that if you do that, all the developmental signals that are communicating between the different cells are close enough between chick and quail that they can interpret that genetically different material can still interpret the same signals. And it turns out what you can do is you can make so you can transplant, depending on which part of the brainstem you're going for, you can basically, I didn't say what happens after you grow this up. So after you grow this up, if you've got a chicken, so here's a chicken, you'll see that there's actually a little bit of quail in it, and there'll be like a little segment of darker feathers which extend all the way up to the skin, because you're taking other kinds of tissue than just brain tissue. And so you can sort of tell, how, that this worked. You can actually see that there's a little bit of quail in that chicken. And so what you can do is you can transplant different behaviors. So you can end up with, like a quail that does, the quail head movement, but chicken sound. And you can do the reverse. You can end up with a chicken that does the chicken's sound, but a quail head movement when it's making the sound. So that shows that very early in development, these cells have already differentiated enough, even though they don't even have very many connections to each other yet. They're differentiated enough that they already know that they're going to make a circuit that makes the quail head movement. And if you transplant that into a chicken, you can actually cause the chicken to make a quail head movement. So the point of this is just that a lot of these signals are a lot more general than you might think. The fact that you can actually, imagine doing this. Like I said, imagine doing this with a computer, like taking like 3/4 of a video card out and just sort of slapping it into another computer. Is that going to work? No way. Everything down to the last transistor has to basically be working in order for a circuit like that to work. Here you can sort of just randomly take some junk and put it in there and then it works. So there's a lot of adaptation and sort of careful adjustment and sort of social studies going on, because cells are talking to each other and getting signals back and forth, because you're assembling this by the group activity of a bunch of intelligent agents. Each cell is an intelligent agent. Okay, so that was one experiment, and I've got some. If you look on the notes, you can see some pictures of some of the things I'm going to talk about. So you can also, in particular, this one. So this one is called ectopic, means like it's in the wrong place. Ectopic fibroblast growth factor 8. Source implant. So the idea is, what are some of the signals that actually specify where cortical areas are? So if you look at a normal mouse, what you see, if you look at it from the top, here's the cortex, there's the olfactory bulbs up there, here's the cortex, and we've got so we're looking at it from the top, and so in a mouse you can sort of, so the cerebellum would be back here. There's a cerebellum back here. So if you look at if you look at the somatosensory cortex, what you see is basically these things called the barrel fields. And the barrel fields are basically a representation of each whisker on the mouth of a rat. And if you look, you'll find little barrels for the feet also. There's a very nice picture in the readings that you can look at. So basically each individual whisker has its own little representation in the cortex. So that's what it's supposed to look like in the normal animal. And we know that there's toward the front of the brain there's a fibroblast growth factor 8. So you can see that was originally isolated from fibroblasts, which are sort of cells like connective-like tissue cells that are growing. And it was growth factor 8. So there's a source of that, and that sort of is one of the things, one of several genes that actually controls the coordinate system. And so what enterprising neurobiologists did was they implant ectopic source. So you leave the real one in there, but just then implant another source. And so what does that look like? So that looks like so here's the brain again. And there's the cerebellum back there. And so you basically have two sources. So you've got the normal source up here, but you can stick another one in back here early in development. And then what happens is you still end up with this representation of all the whiskers on the snout, and then little footies over there. But you can see then you end up with another smaller sort of mirror image version of that, and so you can see the same whiskers. And when you look at that, you can see it really is a mirror image. You kind of mirror image that thing. And what's the relevance of sort of that mirror image business? Well, if you look at many cortical areas, like say V1, cortical area V1 and V2. So V2 is a visual area, visual 2, which is a mirror image of V1. If you look at the somatosensory cortex, if you go out laterally, there's actually a mirror image. This one's called S1, so you should label that. So this one's called somatosensory 1, but there's actually a somatosensory 2 off to the side that's a mirror image of somatosensory 1. And so we think that seems to be one of the ways in which cortical areas have duplicated to form these sort of reversing mirror images. And there's probably a number of other growth factors and signals that we haven't figured out in the cortex, but this gives us some idea that this one at least can cause this sort of mirror image duplication of a cortical area, which is something that we've seen a lot of when you actually look at different animals. So that's you can see it in the auditory system, like there's mirror image duplications of representations of frequencies, there's mirror image duplications of representations of the visual field, mirror images of representation of the body. If you're in a catfish, there's mirror image representations of the taste taste buds that cover the body. So so that's a very general sort of thing. So the last quick and dirty cruise through a series of experiments. So some of these experiments, they're a little Frankenstein. They're kind of Frankenstein, because you're somehow perturbing development to try to figure out how it works. So by adding things that shouldn't be there or mixing and matching between different species. But here's another one that just has to do with a particular gene. So there's a gene called tailless. And you can sort of imagine that has something to do with the back. And it's an old gene. So in a fly, this gene is called, the actual sort of official name of this gene is TLL. And in a mouse, it's just called TLX. So if you want to look up, you got to know the gene names, TLX. So that's the sort of code name for this gene. So tailless, it doesn't sound good, yeah, tailless, you don't have a tail. That's probably not the best plan. But if we go back to our mouse here, so in this case we'll talk about visual areas. So here's a mouse brain, the normal mouse brain, and a normal. So this guy, if you look at this one, there's a big area back here called V1. And so this V1, that's the first visual area. And there's some areas alongside of it like that are higher visual areas. But that's, and then somatosensory cortex, would be, somatosensory cortex is pretty close. It's right about something like this. So here's somatosensory. So there's all little barrels. So that's the normal one when tailless. So you would call this tailless plus because tailless is working. So what happens if you somehow mess with tailless? So there's several different things that you're going to figure it's going to do something to the back. So I mean, tailless obviously it doesn't have a tail, so it's gotten rid of something at the back. But when you actually look at how development works, because it's so interactive, if you take something like the retina going to the tectum, the superior colliculus, in a frog, if here's the retina and it's going to the superior colliculus, and there's a map, so that means like this guy will go here, and this guy will go next to it, and this guy will go maybe over here. So if you have a map, and if you do something like remove part of the retina, so if you remove part of the retina, what will happen is the remaining part of the retina will fill up the entire tectum. So this is the retina and tectum. It'll adjust. It'll figure out, this part's missing, and so what I'm going to do is I'm going to take the map and expand the map out to the part that this part used to project to. Same thing happens in the tectum. So you could take out a piece of the tectum. If you cut out a piece of the tectum, what will happen, and don't mess with the retina, the entire retina will go to the remaining tectum and make a nice map in there. So that's a common thing that happens because cells are interactive, and they're sort of like, they know they're going to make a map in this structure, and if there's not enough of it there, they'll just sort of like, adapt to it. But what happens in tailless is not like that. So what happens in tailless is you get a really short brain. So you end up with this sort of stumpy brain. And if you look in that brain, you can see basically it's got a pretty much normal-sized S1 in it. And then it has this teeny tiny little V1, barely visible. So it's kind of like you just really, cut off the back of the brain. You cut off like the compartments at the back of the brain. And when you look at what happens, when you look at what happens in flies, pretty much a lot of the segments, the back segments just disappear. And so it's not adjusting. So this is a very early gene that's involved in kind of specifying the compartments in the brain, basically, something more like the compartments in the brain. So the point of going over this is not to sort of say that we really understand how this all works. We just have like a few quick and dirty, experiments where you've kind of figured out some of the signals that are going on, but there's probably about another 150 that we haven't gotten to in order to actually specify all these different cortical areas. There might be 10, even in a mouse there's like 10 or 10 or 12 visual areas, and so each one of those appear reliably in every mouse. And so, how did they get there? How did they get sort of duplicated? There's a lot of work remaining to be done, but this just gives you some idea that we have put our hands on, for better or for worse, on some of the, this is the little tiny V1. So we've put our hands on some of the signals that are being used in these systems to actually communicate the structure, sort of the layout, sort of the architectural layout of these things. Okay, so that was some quick and dirty developmental experiments. Okay, so what I'm going to do now is turn over to brain size and allometry, which means kind of how things change as you scale something up or down, and how the measurements change. So it just means sort of other measure. and then finally, we'll talk about how does this relate to cortical folding. So one of the things when you actually look at brains, what you see is that they're incredibly different sizes. So if you take something like, so in order to, and one of the problems with this is that there's certain kinds of sort of almost moral feelings, like, humans have a big brain, we sort of reign over animals, we can sort of like do whatever we want, and animals aren't quite as good, and they've got smaller brains. And so you have this feeling that sort of bigger is better, and since we have a very big brain, then we must be better. So just to get rid of all that sort of kind of sociological underlay, let's just look at animals that are more similar to each other. So let's take like a white-footed mouse. So a little white-footed mouse, here's a little white-footed mouse, got a fourth ventricle, and there's the brain of a white-footed mouse. White, it's a little tiny sort of mice that you find in the field. It's got a little tiny brain. But, you can find other white-footed mice, raised litters, and let's compare that with something like a capybara. This is my go-to example. So what's a capybara? So a white-footed mouse is a small mouse. Capybara is another rodent. It's a different group of rodents. It belongs in the guinea pigs, because you basically have sort of guinea pigs, squirrels, and mice and rats. So capybara is a guinea pig. It's a 150-pound guinea pig. So it's a big honking 150-pound guinea pig. If you've ever had a pet guinea pig, it's just like one of those, except it weighs 150 pounds. And so if you look at this, they've got a huge brain. It's got, there's the olfactory bulb. And if you look at the brain, it's all folded up. It's got a nice, gyrified brain. It's got a huge cerebellum. So, how big is it? Well, it's about 35 times as many neurons. In the cerebellum, it's got 50 times as many neurons, 35 times neurons. So when we compare something like a human and a chimpanzee, we're 2.5 times as big as a chimpanzee. So that's like a factor of 2.5. It's not a huge difference. This is 35 times. So the white-footed mouse brain would basically fit into the olfactory bulb of a capybara. Now, when you look at a capybara, is the capybara sort of cognitively radically different than a white-footed mouse? I think you would be sort of hard-pressed to say that. I mean, capybaras, they can hold their breath and they're good swimmers, and they herd, unlike mice. So they're the only herding rodent, kind of like ungulates. But cognitively, they don't look remarkably, they don't look 35 times smarter than a white-footed mouse. The white-footed mice are pretty smart. They can sort of like, get in your house, and they're hard to catch, and they're really fast. So one of the things we should sort of consider is we don't really understand the effects of brain size. So you have these. So when you look at sort of the brain size, what you see is that maybe 95% of the variance in brain size is explained by body size. True in humans too. By body size. So you actually see that, and you see this like, so if you get a big human, a big heavy human, their brain is going to be bigger. Female brains on average, you'll have large females, small females, but female brains, since they have smaller bodies, are going to have smaller brains. But like the average is like maybe about a 10% difference. So there's this overwhelming effect of body size. Now, when people first thought about this, they thought, well, could this be something having to do with the fact that when you have a bigger body, you need more stuff, you need more neurons to surface it because you got more surface area on a bigger body. Like white-footed mouse just has a couple square inches, but capybara has square feet of skin. It turns out that doesn't work. So when you look at a white-footed mouse, if we flatten out the cortex of a white-footed mouse, what does it look like? Well, it looks like, say this is S1. So this is S1, so that first somatosensory area. So S1 takes up, about a third of the, something like a third of the cortex. So this is total cortex. And there's S1. So what happens if we look at a capybara? Well, a capybara has got like an enormous cortex by comparison, but if you look at the size of S1, it expands some. So here's S1 in a capybara, but what you see is that there's extra secondary cortex, so extra higher level cortex. We'll put it in quotes. So it looks like what happens, and you can see this if you compare primates of different sizes, mice of different sizes. It's just a general phenomenon. So what happens is when you blow up the brain by blowing up the body, what ends up happening is you get extra higher level cortex because we don't need that much. We don't need that much to service the body. And so you always see this tendency for larger brained animals to have more non-primary cortex. So we could say that larger animals have always have larger body size animals always have more non-primary cortex. And this doesn't clearly correlate with, so larger animals aren't always remarkably smaller or have obviously different cognitive abilities. And it's particularly remarkable when you're talking about like 35 times bigger. So the bottom line there is we just don't really understand exactly what brain size means. So in some cases, you can compare two closely related animals and then say one is like a squirrel is very good at vision and one like a rat not so good at vision. In that case, you see the visual areas like V1 is like, say, twice or three times as big in a squirrel. A factor of 2 or something like that. But what about capybaras? Their V1 is like 35 times bigger than a white-footed mouse. And their vision doesn't seem to be 35 times better. They're not super visual animals compared to a white-footed mice are pretty good. And so there's the problem. You can sort of see brain size effects that seem to be related to behavioral adaptations, like squirrels can see better, have color vision than mice. But then you have these things like 35 times more neurons, and you don't see like some radical difference. So I would say the bottom line is we don't really fully understand, how brains can scale and have remarkably number of different, a remarkably large difference in the number of neurons without wildly affecting the functional layout, even when you're doing something like this, by giving an animal a bunch of extra higher level cortex. So it's kind of like if your body gets bigger, you're going to get some extra higher level cortex, whether you need it or not. It's almost that ridiculous. So let's now talk about what happens when you scale up. So one of the things that you saw when you scaled up is you saw that this guy got folded. So how does folding work? So the original theory, one of the original theories of how the cortex folds was, let me erase this down to here, was that perhaps you get gyri where you've got two areas, so gyri meaning sticking out, where you got two areas that need to be connected to each other. And this has sometimes been associated with David Van Essen. So David Van Essen worked on visual areas, and so if you look at the cortex, what you see is that it looks something like this. So here's the cortex at the back of an actual primate brain, a monkey brain, and what you can see is that there's here's area V1, and here's the boundary, and then this is area V2. And when you look at the organization of the map in these two areas, what you see is they're it looks like they're kind of conveniently set up, because this part of the map relates to this part of the map in V2. And so you can see there's all the connections between V1 and V2 to line up their maps, are because their maps are mirror image. So if you have two mirror image maps and you fold the cortex like that, then you can connect them up by just sort of putting little wires that go between corresponding parts. And that's the way the wires actually look like. And so one idea was that perhaps what's going on is that there's tension, the axons that connect up areas, and there's other examples. So if you look at, say, here's area 3B in somatosensory cortex, 3B in area 1 and area 2. You can sort of see the same thing happening there, where there's connections that go across to, say, area 2 like that. So that gave rise to the theory that there's some kind of tension that's actually involved in causing cortical layers to fold up. But then people started doing some experiments, very simple experiments. And so here's an example of an experiment. Say you got a sulcus like this. What if we go down here and make a little cut? So you could just make a little cut in the cortex and see what happens. And so turns out what happens is that there's some stress on the cortex this way. And so what happens is this cut will open up in that direction. So that's a little sort of counterintuitive. Why is there stress down here? That's away from the place where there ought to be tension. So these experiments, if you want to look, there's experiments done by this experimenter G. These were done in 2010. I have some of the references in the reading list if you want to look at this. But then they did something directly to test this idea. And so this is a really good example. You came up with an idea. It probably is not right, but it doesn't matter. It actually sort of motivated a test. And so what the test was, let's take a gyrus. So here's a gyrus. So you might expect if you put a cut like that across the gyrus, you're expecting there to be tension sort of across the gyrus here in this axis, because of these guys are sort of tensioning it up and causing the folds to happen. But if you do that and see what happens, nothing happens. It doesn't open up. But conversely, if you actually make a cut like this, oh, sorry, I did it wrong way. Yeah. How silly of me. So, in fact, it opens up this way. So you see that there's actually some tension in the gyrus, and that's kind of holding the gyrus down from the top. Whereas if you make this kind of cut, sorry, I did it backwards. If you make this kind of cut, which is the one that Van Essen would predict you to have tension between V1 and V2, what happens instead is nothing. So when you do that cut, it doesn't open up. And so that suggests that there isn't any tension across this way. And in fact, the tension is along this axis. It's almost like the gyrus kind of pulled up away from the white matter, and that created some tension. Exactly the reverse of this prediction. And so then I put another nice reading in the there's a guy and he did some sort of computer models, but also physical models. And so what he started off with is let's just make a little armature, a hard armature of the white matter. So this is kind of the white matter underneath the cortex, and then coat that with a gel. So here's like a little gel on top of that. So this is a gel, and then this is the gel that when you expose it to water, it expands. And why is that a model of the cortex? Well, as we'll see when we talk about the subplate expands a lot. The white matter expands a little bit less than the subplate does. And so if you just make this very simple model, which is just basically make a white matter shaped armature, coat it with a thin gel, allow the gel to expand, then what happens? Well, amazingly, you get a lot of the traditional folds that we associate with the primate brain, because this is the primate shaped white matter. And in particular, what you see is and you can actually do this with modeling studies, or actually just trying to measure the direct stress in this thing. And what you see is that there's a huge amount of compressive stress. So in these places where you see some of the major sulci, like the central sulcus and the superior temporal sulcus, you have a max in compressive stress, and that actually corresponds to where the folds appear in this completely sort of physical model of the folding. So what's actually going on? So the way to think of it is let's just do it as a sphere. So you start off with a little layer on top of the sphere like that, and then what happens if you expand the outer layer but not the hard inner layer? What's going to happen is you'll get sort of things thrown up into folds like that. And so what would you expect there? You'd expect there to be stress in this direction, because those are like we saw over here, because that gyrus is kind of pulled away from the underlying white matter. and then you wouldn't see stress in this way, and then you would see sort of compressive stress helping you fold the cortex. So what's actually causing the cortex to fold isn't the axons connecting it, it's this buildup in compressive stress from the gel kind of expanding, the gel of the cortex expanding and sort of compressing its neighbors. So you can look at those papers in the reading, but that's probably the physical explanation for the gyrification. When you look at the gyruses in an animal like capybara, because it has a differently shaped white matter, the layout of the sulci is quite different than it is in a primate where you have a big temporal lobe. So that's sort of an overview of what we think is actually sort of causing the folds to occur when you've got a really big brain. And so if you look at a primate like an owl monkey with a very small body, it only weighs like about 2 pounds, it has a smooth brain. Is it a dumb monkey? No, it's just looks like a smart enough monkey. Monkeys around. And when you look at an animal like a baboon with like a 70-pound body, it has a much more folded brain. Looks even more like a human brain than a macaque monkey because its body is so much bigger. You start to see some of the same folds that you see in humans appearing. So, okay, so that was a quick and dirty summary of where do we think the folding pattern comes from. And you can use the folding pattern to line up brains, but it's important to get some idea of it's probably not directly related to cortical areas. It may be the case that sometimes you have a thinner cortical area and that tends to fold a little bit, but in general, we would have to say that it's probably more due to the overall shape of the brain with a thin cortical layer on top that's expanding relative to the armature it's on. That's what's causing folds to happen. Okay, so now let's completely change over and talk about the subplate. And so to talk about the subplate, we need a little bit of a background about cortical evolution. So before the subplate, if you look at a reptile, what did the cortex look like? And so if you look at a reptile like a turtle, if you look up in the forebrain, you have a cortex. So there's something called dorsal cortex in the turtle. And then there's another little thing sticking out over here. And then you got the striatum, because remember, and then there's the septum. Don't forget about the septum. So here's the septum. So that's a picture of the forebrain of a turtle. And so what are the parts there? So this guy is called the dorsal cortex. And this part, dorsal cortex goes to about here. This part is called the dorsal ventricular ridge. Dorsal, we'll see something like that in a mammalian embryo. Dorsal ventricular ridge. It's not cortical particularly. And then this part's the striatum. So remember, these parts right here are the pallium. So if we go back to our original picture of the forebrain, very simple picture as just 2 tubes like this. Remember, we had the septum over here, and then the basal forebrain over here, and then the pallium on top. So the dorsal-ventricular ridge and the dorsal cortex are both part of the pallium, and then here's the striatum, which is we can call that just striatum here. Striatum, and then this guy is the septum here. Okay, so what does the dorsal cortex look like in a turtle? So it's got It has pyramidal neurons in it, so here's the pyramidal neurons in it. But if you blow it up, what it looks like, it looks kind of different than a regular old mammalian cortex. So what it looks like is you just have one layer of cells. So here's these cells, and they send up multiple apical dendrites, and they've got spines on them, just like cortical cells are supposed to have. And so there's this one main layer of cells, and there's a few inhibitory cells interspersed up here. So here's like inhibitory. But basically, there's just basically one layer of cells, whereas the cortex in a mammal is a thick thing. And then here's the ventricle down here. That's the same ventricle, the original ventricle. And when we look at how the connections come in, if you look at the dorsolateral geniculate, so this guy has a map in it, map of the retina in a turtle. So map of the retina. So that guy has a map of the retina. But when the cells in there come in, they come into the dorsal cortex and they just go across in lines, and they synapse. There's no map in the cortex. They just come up and they make synapses all along. There's no map. Now, if you look at that kind of a structure, if you look at the olfactory cortex looks like that. And so that's partly what gave rise to the incorrect idea that there was a smell brain. So it was never a smell brain. It was always visual cortex. It just happens that the structure that you see in olfactory cortex was more widespread. So it isn't that you started off with a smell brain. It could easily be that you started off with this thing because this kind of a structure is what you need for the visual cortex, and you also used it for the olfactory cortex. So you can also see something that looks like that if you look in the hippocampus. But you still can see something like this if you look throughout the entire cortex of a mammal in layer 1. So layer 1 looks just like this. So if you look at layer 1, there's hardly any cells in it. There's dendrites sticking up in there with spines on them, and you've got axons that come from different cortical areas, and they run sideways and contact large numbers of different cells very weakly. So you have exactly the same structure at layer 1, of all mammalian cortex. So it's still there. So this kind of structure is still there. So you still have a smell. If you had a smell brain before, you still have a smell brain. It's still there. So when you look at mammals, one of the things that really characterizes mammals compared to reptiles is that they have all these maps in the cortex. So they've got these maps of different. You have maps of the visual system, maps of the auditory system. We'll talk a lot about that when we get to the visual system. So they've got maps, visual, auditory, somatosensory maps. And so how do those maps in the cortex come about? Now, reptiles have plenty of maps. They have maps down in the superior colliculus, and they have motor maps. And so it's not that all maps emerged in mammals. There's maps all over the place already. But the particular kind of maps that we have in the mammalian cortex seem to be involved with this thing called the formation of the subplate. And so what is the subplate? So we'll start off with, and so what part of development we're talking about here? So I'm going to talk about all embryonic development. So we'll start off with our so I'll put 4 stages across here. So the pre-subplate. So what does subplate mean? Well, plate just means the eventual completed cortex. And so This is an embryonic, this is what I'm drawing here is a macaque, so it's embryonic day 50, so 50 days after conception, which in a human equals about what did I put, 84, about 84 days. So 50 days, that's what that, so 84 days for a human. And then now we have subplate early here, and that would be about 70 days, and in a human that equals about 130 days, because human gestation is slower. And then subplate late, and that's 90 days, and a human equals about 180. And then subplate dissolution. So that's, subplate sort of disappears, and that's about E120, human equals 245. And then gestation, the actual birth. And that's going to be, there's birth, yeah. So that's 165 days in a macaque monkey, and human equals 280. Okay, so we've got that set up. So let's start off with, on the right side here, all the different zones we're going to talk about. So we have the marginal zone. And the marginal zone is basically layer 1. And then we have the cortical plate. And that's normally what you would think of as, the cortex. And then we have the star of the show, which is the subplate. So something that sort of is underneath the cortical plate. And then there's the well-named intermediate zone. And then the one that takes the cake for the most ridiculous name, the subventricular zone. And why is that stupid? Because the ventricular zone is underneath it. So why didn't they call it the supraventricular zone? So the ventricular zone is where the cells are dividing. It turns out there's some cells dividing in the ventricular zone too. So we'll look at how these set of layers come and go. So there's, how many of them? 6. Okay, so if we look at the pre-subplate, what happens is you already have, if we draw sort of a section through the cortex, you've already got some plate, meaning like, final cortex. And so what does that look like? So we've already got some cortical plate happening. And so that part is very dense with cells. So those are cells that are post-mitotic cells, and they've actually sort of now become the beginning of the cortex. It's very thin. And so what does that correspond to? So that's the plate, cortical plate. So that guy's there. And then here's the layer 1. So layer 1 is characterized because it's got a lot less cells in it. So that's layer 1. So that's going to turn into layer 1. And then we've got very dense cells down here that are the ones right along the ventricle that climb up and down the guy wires and divide. So that's the ventricular zone. So that's the ventricular zone, and then there isn't much of a intermediate zone to speak of. There's just sort of a few scattered cells. So we don't exactly know where the different layers are. So we just you just have 2 basic layers of cells: the ones that are dividing, and then the ones that are post-mitotic. So that's an early And that's kind of the way I thought of the brain and before I actually read about this. I just thought of, there's some dividing cells down here, and then there's some cells in the cortex. But it turns out when you look at a little bit later in development, you've got this absolutely gigantic column of cells. And when I first heard about the subplate, I thought, yeah, it's some little cells down there. It's not some little cells down there. In a human, it's like 6 times thicker than the cortex. It's this giant plate of cells, the subplate. And so if you look a little bit later on, what you see is, the cortical plate has gotten a little thicker. So now this is the thing that's going to be the final cortex. So we'll just sort of focus on that one. So that one's a little thicker. Very densely packed cells in there. They're all done post-mitotic. So that's so this guy sort of corresponds to that. It got a little thicker. Layer 1 is still there, and it's not quite that thick. We'll make it a little thinner. So layer 1 has a small number of cells in it. They're still there. But then where all the action is going on is down in the subplate, and so the subplate is this absolutely unbelievably complicated thing. It's got little sort of columns in it. It's got little sublayers in it. And then there's some subventricular zone. And then here's our, ventricular zone. So here's the ventricular zone down here, but there's some cells dividing above the ventricular zone. So there's cells not just in the really dense ventricular zone that are dividing, but there's some dividing there too. And so here's that ventricular zone down here, but then there's kind of a subventricular zone. And then there's an intermediate zone. So here's the intermediate zone, something like that. And then this huge thing here, which had almost nothing in it over here, is now completely filled with cells. So this guy is the subplate. And like I said, it gets particularly big in a human. And it's got a lot of complexity in it, lots of different cells in there, little layers that come and go. And so what's the relevance of this? Well, it turns out that if you look at the cells that are actually connecting up, say like the dorsolateral geniculate nucleus. So that's the dorsolateral geniculate nucleus. Has a visual map in it that comes from the retina, and it projects up into the cortex. Well, it turns out the subplate's absolutely critical for it, because what happens is these cells come up and they grow up and they actually connect to cells that are actually in the subplate. That's not their final connection. So they actually make temporary connections in there, and you can even see something like there's also electrical synapses in there. So electrical synapses. I didn't even mention those electrical synapses. So what happens is if you have two cells, there are these certain proteins that are they're like a little grommet. It's like a little grommet like this. It sort of like fits across two cells in order to electrically connect them. And so then instead of a synapse, normally in a synapse you release some neurotransmitter, it opens a channel, some ions flowing through the channel. But here the ions can actually go directly through, just directly through a connection between the cells. So that happens in the subplate, so they're really intimately connected. You can tell if you inject. How do you tell this? Well, inject a little dye into this guy, and you'll see the dye will sort of spread into several cells. So these cells they make electrical synapses. Synapses, regular chemical synapses, happen onto them from the dorsolateral geniculate. So there's a whole bunch of stuff going on. If you mess with any of this, you won't get a visual map. So it's known that if you disturb the subplate, it's important for the visual map. But what happens after that, and how do these cells get up here into the the regular cortical plate. And so they get up there by basically migrating. So this dotted line means that it's crawling. So it crawls, and what it does is it crawls through all the cells there and starts piling up on the top. And so that means that the oldest born cells are here, and here's the youngest. So the cortex is kind of inside out. So the younger, the more recently birthed cells are on the top of the cortex. So the lower layers of the cortex are the oldest cells. So then what happens is the cortical plate, gets bigger. So here's the cortical plate. Now it's gotten bigger, and then the cortical. So this is like all incredibly dense cells here, and then the cortical plate, the subplate, starts to actually disintegrate. So you see some of the layering goes away, and that happens after the cells, after these cells that are in the subplate have actually migrated into the plate proper. So it's a complicated thing. It's not very well understood. I mean, when I first heard about this, I thought, well, yeah, the subplate, I thought it was just like a little thing. I didn't realize it's this giant thing bigger than the cortex, and that goes away. So now we have lots of cells in the plate, and there's less of them. There's still some layers down in here in the subventricular zone, but this basically is so here's the ventricular zone right there. Subventricular zone is there. Here's those ones that are harder to tell where you are, and then here's the plate, which is getting thicker, and there's layer 1, which is getting thinner. And so when you finally get up to the subplate completely going away, then you have a much thicker, cortex. So this is the basically layer 1 of the cortex here, layer 1, layer 6 down here. So you have all, and you start to get actual cells. They start to differentiate. So here's some pyramidal cells, in there. There's pyramidal cells at different layers. That's what the little dots are supposed to represent, those cells. They've actually started to differentiate. And there's just a few cells in the top, in layer 1. And this is basically the white matter. So pretty much all the cells go away. So actually in the space that used to be white matter, that's actually where the subplate, all these cells in the subplate were. And then you still have the ventricular zone here, and the subventricular zone kind of goes away. So the ventricular zone can still make cells. So if you look at sort of like a woman during the menstrual cycle, the hormonal stimulus during the menstrual cycle will cause some of these cells that are near the hippocampus to divide and actually generate new neurons, put new neurons into the hippocampus. And the same thing happens in birds for the birdsong nuclei. So in the adult animal, it's still possible to make neurons in just a small number of places. It's rare in general. So there's layer 1, and then these additional subventricular intermediate zone pretty much sort of go away. And you just end up with a white matter. It doesn't really have any cells in it anymore, any neurons in it. So that's your sort of adult cortex. So last but not least, what I wanted to talk about was two things. So one of the things I wanted to talk about is this idea. There's been an idea for a long time that a particular portion of the ventricular zone is responsible for a column of cells. So basically, you can sort of make a nice column of cells here. So you see a lot of columnar structure in the cortex. And so the cell that initially divided in the ventricular to subventricular zone will go up and end up at sort of the same place in the map defined by this ventricular zone surface. And in general, that's true. So you can see that pyramidal neurons that were over here came from this part of the ventricular zone. Pyramidal neurons that were over here came from this part. But it turns out that if you look at the inhibitory cells, they came from somewhere else. And so if you look at an embryonic mammalian brain, it kind of looks like a turtle. And so here's the ventricular zone, and it's causing pyramidal neurons. Here's some pyramidal neurons. So, the cells that divided here went up and formed pyramidal neurons here. But it turns out that the inhibitory cells actually come from down here. This thing is called the ganglionic, in a mammal, it's called the ganglionic eminence. So So the inhibitory cells are over here, and they actually just crawl sideways through the cortex. And then when they get to wherever they want to be, then they sort of go up in the cortex. So they're actually generated from the ventricular zone down here, and then they crawl sideways into the cortex, whereas the pyramidal neurons are generated in place at the same part of the ventricular zone and strictly sort of grow upward. So that was something that was discovered about 20, 30 years ago. That's really completely changed our idea of how the different cell types get in there. So the inhibitory cells come from a different area, whereas the pyramidal cells kind of almost more like define a particular area. But the last thing I wanted to talk about was I'll just talk about the Wheeler mouse. And so the Wheeler mouse was discovered. It's a mutant that was discovered a long time ago, 1951. And so one of the ways to study, genetics in the early days was we just look for mutants. You could irradiate, a mouse, do bad things to a mouse, and you get a mutant. So the Wheeler mouse, as you might imagine, the Wheeler mouse has a little trouble getting around. It's partly because of some problems in the cerebellum, so it has a tendency to not walk completely straight. And it was discovered that there's a protein that was named after the Wheeler mouse called relin, and it's in these cells that Cahal had seen. They're called Cahal retzius cells in layer 1. So these guys are in layer 1. And so if relin gets messed up, there's no relin in these Cahal retzius cells. There's like CR here. So they're up in layer 1. And so then what happens? Well, the most amazing thing happens, and this is a great illustration of how kind of the intelligent agents that assemble the nervous system, can actually sort of figure stuff out. So what happens is the cortical plate, so not the subplate, but the cortical plate, piles up below the subplate. So what does that mean? That means, so normally what's happening here, you've got this giant subplate thing here, and then here's the plate. So the axons come in, they attach to cells in the subplate, and then eventually the cells in the subplate migrate up into the plate, and the connections break with them down here, and then they actually go up and connect to cells in the plate. So what's happening in the Wheeler instead is that in between the ventricular zone and the plate, the ventricular zone and the subplate, this whole sort of final cortical plate thing is stuck here, completely in the wrong place. And so how does that sort of mess up this problem where the thalamic cells have to go and connect up to the subplate cells first and then disconnect from them? The cells go up into the plate and they reconnect to them up there. Well, it still does it. So what happens is, thalamic cells crawl, thalamic cell axons crawl through the plate. So they crawl through the thing that they're going to eventually connect up to. This thing, which has like strangely gotten, displaced down to here. So this is realer. So they actually crawl through the thing that they're going to eventually attach up to in order to get to the subplate, because they have to get to the subplate and talk to the guys in the subplate, connect up to them. So they crawl through the subplate, and then once they've crawled through the subplate, they then crawl back to connect up to the plate. So then, crawl, down to plate to make their final connection. And what you get is pretty much, a more or less normal mouse. It's not, it's a little fidgety, but basically it's a mouse. It can mate with other mice, raise a litter. So it's just unbelievable. You've got everything completely inverted. And, the way that the cells in the plate sort of crawl, like the oldest ones are here and the newer ones crawl through the plate and then pile up at the top, that's reversed also, or sort of completely messed up. And so you can't see any of the normal cortical layers. The cortical layers are all sort of disarranged in realer mouse, and yet the connections are there. The geniculate map is there. They still have multiple cortical areas. They can perceive other mice, and so it's just it's interesting. You have this weird mix of this incredible ability to adapt to completely messed up developmental rules, but sometimes you have things like tailless, where you just chopped off all the back segments in the brain. So you have sort of a combination of those two. And this is just kind of like a quick and dirty overview. We don't really understand how all of this works. It's relatively hard to study this because this is all in an embryo. And so the only way you can study this is you have to, if you want to disturb this somehow, you'd have to take a fetus out, do some modification to it, and put it back in. And those kinds of experiments have actually been done. But it's obviously something that's actually quite hard to study. And so not much is known about this, but we do know that if you disturb anything with the subplate, it disturbs all the eventual cortical maps. And since mammals have all these cortical maps and reptiles don't, and they don't have the same sort of subplate setup, we think that this whole system must be somehow involved in the creation of cortical maps. Okay, so I went over a little bit, but I think we'll end the recording now.